How's everybody? Is it shocking to be here? Back from break? 2, 4, 6, 8, 10, 12. Not bad. What is that? 75% or so. Okay. So last time we had started by talking about saccharides. And let me get the timer going. And it occurs to me that word, who's heard the word saccharide before? Outside of this class, a majority of you. Maybe you've heard the word monosaccharide. Maybe you've heard the word disaccharide or polysaccharide. Here are just some definitions of them, so you can get the broader landscape of sugars, saccharides, carbohydrates. These are all synonymous, fairly synonymous. I'm going to give you the sort of overarching landscape of saccharides, and this is going to be the map that we're going to walk along in this chapter, all right, this unit about saccharides. So first, we're going to talk about monosaccharides. And this, when you say sugar, this is usually what you're talking about, either a mono or a disaccharide. A simple sugar is a monosaccharide. So last time I had shown you the tree of the aldoses, that tree of sugars where the carbon chain was growing. Those are all monosaccharides. So there are about 70 or so monosaccharides, either ketoses or aldoses, that are D. There's actually 140 if you consider L sugars. And of those, about 20 are found in nature. There may be a few more than that in some weird bacteria somewhere that grows in a thermal vent, but that's the majority of them. What are some examples? Well, glucose, blood sugar, and fructose are two simple sugars that we're going to talk about quite a bit today and on Wednesday. And so this is what most people think of when they think of sugars. It's these, like this candy right here, is mostly made out of some kind of simple sugar. Next is disaccharides. So a disaccharide is a molecule made by the formation of an ether linkage, or what's called a glycosidic linkage, between two monosaccharides. So you're going to have two simple sugars linked together to form a disaccharide. So an example of that, the most commonly recognized example, at least by people, is sucrose. You've probably heard sucrose. Sucrose is table sugar, some kind of a disaccharide. We'll see what that looks like. In fact, it's a disaccharide that combines glucose and fructose into a single molecule. And so but there's a lot of different types of disaccharides, and this is sort of the realm that you're going to find them in here. You'll find them in a lot of the fruits and vegetables and nuts and grains that are out there. That you're going to have examples of disaccharides. Next is polysaccharides. In between is this realm called oligosaccharides. Oligosaccharides are short chains, longer than 2 units, but they are not very long. Polysaccharides includes oligosaccharides, but can also include very long chains. So long chains where each of the subunits is a repeating sugar ring that is linked by this ether or glycosidic linkage that you're going to see in a little bit. Okay, and so there's a lot of examples of these. Often when we talk about fiber, soluble and insoluble fiber, there's a range of those, and that's going to be kind of one of the last topics that I talk about in this lecture series. We'll see that, but some other important ones are vegetable starch is a type of polysaccharide. This is called amylose, And you find this in potatoes and grains, starchy grains, and vegetables like corn and wheat. We've looked at wheat flour under the microscope, and we've seen in the white wheat flour those sort of white snowy clumps. That's starch. Those are starch crystals from the wheat berry. Polysaccharides. All right. So now again, we're gonna get into the structure of sugars a little bit, and this is like such quicksand for me for a number of reasons. Number one, I've already told you guys that this is my area of research, so I love this, and I realize that most people don't necessarily love this. But to talk about the structure of sugars, I do want you guys to have some sense of what you're looking at. So I'm going to try to give a little bit of what's called structural glycobiology, where we look at the structure of these sugars. And to do that, I'm going to give you just a little bit of background. All right, so when you look at any carbon center, a carbon is often tetrahedral, meaning it has 4 things attached to it, right? I handed you a tetrahedral molecule last time that it looked like a jack that you may play with or something, right? It had the carbon in the middle and 4 things sticking out of it. Well, a tetrahedron, it projects into space. It's a 3-dimensional object, right? It's a 3-dimensional shape. And how do you represent a 3-dimensional shape on paper? You know, ostensibly a 2-dimensional surface. So this guy, Emil Fischer, was the first to really come up with a way of drawing it. And so he just said, well, when you draw something, I'm going to draw these sugars, and the way I draw them, I'm going to have a linear chain going up and down of carbons and things sticking out from the side. And when I do, I know that the things sticking out of to the sides are pointing towards me, and the things in the chain, the vertical chain, are pointing away from me. All right. So this was his sort of graphical representation of the 3-dimensional structure around a carbon atom, and he used this to be able to describe the stereochemistry of carbohydrates and what has now become called the Fischer projection. Yeah. So stereochemistry refers to, thank you for asking, next point, is simply the idea that a molecule has a 3-dimensional shape. All right, so stereo, when you're listening to music in stereo, or you have stereoscopic vision, you have like depth perception, like a perception of the 3-dimensionality of the world, or listening to things in stereo, it's giving you a sense of the spatial distribution of sound. The stereochemistry of a molecule is looking at the 3-dimensional structure of that molecule. All right, so the stereochemistry or 3-dimensional arrangement of the atoms in a molecule can be depicted using this Fischer projection, which specifically applies to carbohydrates. All right, because that's what this guy worked on. As I had said, he's like my academic great-great-grandfather or something. Interesting guy, did a lot. We're not going to take the time to dig into his history too much, though. Although it is interesting. So here's the simplest sugar, so to speak. It's a 3-carbon sugar called glyceraldehyde. Glyceraldehyde is a metabolic intermediate in glycolysis in your body. That is the process by which your body utilizes sugar and converts it into energy. So you can take glucose and break it down into simpler molecules, and one of those molecules in that process of breakdown or catabolism is glyceraldehyde. And it is the smallest molecule that we can consider to be a sugar. The way glyceraldehyde looks is it has this aldehyde group, this C double bond O on one end, and then it's got a chain of carbons sticking off of it each of which has a hydroxyl group attached to it. And so when we're using the Fischer projection to look at it, we see that looking at it, we just, without having all the colored boxes around it, just seeing the letters with the lines attached to them, I would be able to uniquely build the structure of glyceraldehyde with its complete stereochemistry. All right. So glyceraldehyde has what's called a chiral center. A chiral center is, you'll remember from last time, I handed out all those things and tried to get you to figure out what the mirror images of, the non-superimposable mirror image around that center. There was a center that had 4 different things sticking out of it, a tetrahedral atom with 4 different molecular centers emerging from it. Glyceraldehyde has a chiral center. If you look at that carbon, in the 4 directions that the bonds point, each of those directions is pointing towards something that's different and unique. All right, and we could have a different arrangement of those. Yep, that's right. So, for example, the hydrogen to the left and the hydroxyl group to the right could be switched, and it would be a non-superimposable mirror image. Because of this chiral center, the molecule is said to be optically active. Oh, I should define the word enantiomer. So chiral means it's going to be optically active, and I'll show you what that means in a moment. Enantiomer is the word that we use to mean non-superimposable mirror image. All right. So, for example, your hands are enantiomers of one another. They're mirror images that you can't superimpose and map point for point onto one. Right. Does that make sense? Not really?
I don't know why.
Another example would be a pair of shoes would be enantiomers. They are mirror images of one another, but they're not superimposable. You can't take one and make it transparent and lay it on top of the other, and they would appear to be identical. You know what I'm saying? They're non-superimposable mirror images. Whereas oranges, 2 oranges are superimposable mirror images of one another. You can take one orange and stick it on top of the other orange, and they'll look the same.
Also, I see the definition.
Yeah, you hang on to your hat. That's the next slide. So, it's optical activity. The definition is the ability of a chiral molecule to rotate the plane of polarized light. So, here's optical activity. Light is a wave, right? And that wave is more like, I think of it almost like beads on a chain. The wave isn't going just up and down, it's going side to side and in all directions along the ray of light. You can take regular normal light and pass it through a polarizing filter, and that's what polarized sunglasses are, for example. And those are essentially like microscopic slits. You can think of them as microscopic slits that are only going to let light that lines up perfectly with those slits pass through, and all the other stuff that is at right angles to those slits gets knocked out, all right, and is not able to pass. That's called polarizing light. So if you take polarizing light that's all traveling in the same direction, and you shine it on a chiral molecule, it's going to deflect that light either left or right. And the amount that it deflects it is dependent on the unique structural properties of a molecule. Chiral enantiomers are going to deflect polarized light equally, but in opposite directions. All right? One is going to rotate it to the left, and the other is going to rotate it to the right. That's what optical activity means. Yeah. Yep. So you can imagine, like if somebody threw a snowball at you and you put your hand up like this to stop the snowball maybe would bounce off this way because your thumb is pointing out this way. It would bounce to the left. Whereas if you put your other hand up, it would bounce to the right, maybe. I mean, I'm just making that up. But you know what I'm saying? That's kind of like chiral enantiomers and optical activity. What the hell does this have to do with food? My God. Okay, I'm getting there. So the thing is, sugars can be either L or D. And I'll tell you in a bit about this really weird thing about the universe and at least life on Earth. All the sugar on Earth is D. Why? You have to hold your breath and wait. I will tell you. But L sugars have the arrangement that you see here in glyceraldehyde, where the hydroxyl is sticking off to the left, and D-glyceraldehyde has it sticking off to the right. So L just means levorotary, meaning rotating polarized light to the left, and dextrorotary just means rotating polarized light to the right. Like dexterous means right-handed or whatever. It's kind of prejudice against left-handed people. All right. So this is optical activity. And when we call a sugar D or L, we are specifically referring to the optical activity or the configuration of that first hydroxyl group next to the carbon, the aldehyde group, the aldehyde or the ketone. All right? We'll see why in a moment. We're going to do an experiment. What? Experiment? This is lecture. Okay. So everyone's going to get to do it. I'm going to give one to you. Here, take that. And one to you. And it's like a wedding. Here comes the bouquet. Somebody grab it. Oh, it's Meredith. And so what you're looking at is one of the things. I took a picture of this at breakfast this morning. So here is a molecule of glucose. This is a molecule of glucose. The little green ball is a hydrogen, just like all the white balls, but I'm labeling it green so that you won't lose track of it because we're going to do something special with it. And it has these weird bendy bonds to it because we're going to do something special with those as well. In fact, the oxygen that has those bendy bonds to it is going to be the oxygen that forms the aldehyde group. All right? And that purple bond is labeled because we're going to specially use that one as well. All right, so here is the one on the left is what is in your hands. The one on the right is actually open chain glucose. And we're going to try to move from the ring form to the open chain form. So what I would like you to do is pull the electrons, and everybody pay attention to this because this group of three people is going to do this, then they're going to hand it off. And the next person is going to do it in reverse. And they're going to hand it off. And they're going to do it in reverse. You're going to keep doing it for a little bit. And then we're going to see what has happened to what we've done. All right? But before we do that, I want you to notice, to really take a good look at the structure, particularly the structure right around of the molecule that's in your hand. Oh, he's going for it already. You did it. Okay, great. Well, so you pull the electrons, the long gray bond away from the hydrogen and the green ball. All right. Next step, you plug the electrons, the purple bond, into the green ball. And then you plug the bendy bond that's still hanging off that oxygen, and you plug it in to the hole that's left in the carbon. All right. You have just made, let's see, that looks pretty good. Yep. Now you can stretch the molecule out and look at the open chain form of glucose. Yeah, you can stretch it out and look at what open chain. So you can rotate some of the bonds once you rotate them. Yeah. There is open chain glucose. All right. And then do that, and you hand it on. Pass it to somebody else. They're going to go in the opposite direction. And we're going to keep this going around the room for a little bit. I want everyone to get at least a chance taking it apart and putting it together, but you only do one at a time. So now, these people are going to unplug one of those bendy bonds. Doesn't matter which, just unplug them. And step 2, unplug the purple bond from the green bond. Yeah. Next step, plug that purple bond into the carbon, the hole in the carbon. Oh, no, Theo, you did it wrong. You got to have the electron. Nope. Stays attached to the oxygen. Oxygens are electronegative. They hold the electrons. Yep, that's right. So plug the purple bond into the carbon. And then, ooh, something weird happened there too. Maybe this experiment. So let's, yeah, one of the mistakes here is that you got to make sure when you're pulling it apart, the last thing that here. Sure. What's that? Falling off. 4 or 5. Yeah, something's gone.
You're not getting it back.
Nope. There should be 6 red balls. There's only 5 there. So an electron. Between the 2 of you, there's an oxygen missing.
Deformed. What would you call that? Does that happen?
That's amazing. Okay. Well, then plug the bendy for the 2 that aren't destroyed there. It's okay. This is cool. It's like there's got to be another oxygen somewhere. Right. That's not it. Yeah, you plug the bendy into the green, and then you pass it on again. Okay, so keep those 2 going around. Let's go over it again, because you pull the purple bond away from the carbon. You're always taking electrons away from carbon and leaving them on the oxygen. There you go. And then you're taking the green off of the purple, sticking it on the bendy, and plugging the purple back into the hole on the carbon. This is a real experiment. Something will happen. Did that work? Yeah, that looks okay. I think. Oh yeah, that looks good. We'll get to it. It is an experiment. There is a purpose to this. Wish I had 3 because it's not going to work as well.
Rotate forward.
I don't know what happened to it. Okay. Okay. Let's move on with it. Keep it going around the circle. Does everybody feel like they understand reasonably well how to do that? Okay, you keep going while I'll get to the point of this experiment in a minute. So, the idea is what you are forming when you take the open chain and plug it back together is a hemiacetal or a hemiketal. So, alcohols and carbonyl groups of an aldehyde can react to form a hemiacetal. Whereas if that carbonyl group is in a ketone, it forms a hemiketal. So to remind you what an aldehyde is, an aldehyde in general is what you see up on the upper right-hand corner there. You're going to have a carbon with a double bond to an oxygen. It's going to be attached, the 3rd bond will be to a hydrogen, and the 4th bond will be to something else. another chain of carbon, the rest of the molecule. Whereas in a ketone, it's the same thing except that there's carbon chains coming out of both sides of it. All right. So sugars can either be aldehydes or ketones. And then you can take those aldehydes or ketones and react them with a hydroxyl group, which we all know, we've seen that sugars are coated with, those OH groups, the alcohol groups. We call them also hydroxyl groups. It forms a hemiacetal or a hemiketal, which is this structure that you see here on the right-hand side. All right? You're handing it to me. And I'll do a little bit of quality control. That looks good. All right, cool. All right, and this is the structure that helps the sugar form a ring. So here are two common hexoses. Glucose, this is alpha glucose, and fructose. The two of them are, the glucose is an aldose because it has an aldehyde, that's the one you're working on, and fructose is a ketose, meaning the carbonyl group in the open chain is actually a ketone. All right, so let's look at these two a little bit and how this forms in each of them. Glucose can open to form an aldehyde. This is essentially the reaction you're doing. You're taking electrons, that little red arrow in the upper right, in the corner there, is electrons flowing from the hydrogen oxygen bond towards that carbon of the carbonyl group. Yeah, that's good. So you pull the hydrogen off the purple bond, pop one of the bendy bonds out of the carbon, and then switch them around. The same chemical reaction happens in glucose and in fructose. You can see here in the glucose in the open chain Fischer representation that the double bond O is at the end of the sugar, whereas in fructose it's the second carbon in. All right, and that second carbon in is how it's referred to as it's why it's referred to as a ketone. And then sugars that have these ketone groups are what we call ketoses. They have slightly different chemistry, and glucose is significant, like the way our body responds to them as taste is significantly different. So when you taste glucose syrup, I don't know if anyone's ever had glucose syrup, but glucose syrup is not that sweet, and fructose is very sweet. Like, so if you've had glucose syrup, yeah, it's not something that's overwhelmingly sweet to your taste. Whereas if you had high fructose corn syrup, you'd find the body, the taste, the way it responds to taste is the response to the chemical structure, the stereochemistry of it, and we perceive that differently. No, because amongst the aldoses, there are a bunch of different shapes as well, and it's not just what is going to be called the anomeric center, which is where this aldehyde or the ketone is. It's the overall structure of the molecule. Yeah, carbohydrates, I'll say this before we get into it too much further. Carbohydrates have long lagged the work that's been done in like proteins because people are daunted by carbohydrates. They're very complex, and and there's a lot of diversity in them, a structural diversity, which is exquisitely, first of all, very dynamically flexible. It changes. Proteins tend to be a little bit more static in their structure. And that dynamic, complex structural, three-dimensional structure of a sugar is almost always related to what its role is in a biological context. And then there aren't very many good tools to explore it. That's the real problem. There aren't a lot of good experimental tools to dive into this structural biology of carbohydrates. I don't want to get too hung up on that. Okay, so this is a little zoom in of what a typical aldose and a ketose would look like in that carbon center that you guys are shuttling back and forth. So is this one in a ring form over there?
Yeah,
No, there's an oxygen in the ring. So, yeah, something went off. How did that one? Would you try to close that one up and we'll see where that one got? So you unplug the purple bond from the green, and you unplug the white ball from the gray bendy one, or the gray. I'm sorry. So that one, yeah, you unplug the bendy one from the black one. Mm-hmm. Yep, either of those. And you put the purple, nope, that's just one. And you put the green on the end of the bendy gray, and then you plug the purple back into the hole in the black. That's it. That's right. Yeah, that one worked. Okay. So, these structures were hard to wrap your head around, and a lot of people tried to come up with different ways of representing them. This guy has the Haworth projection. It's another way of showing a sugar in a ring form. You're going to see a lot of these structures, so I'm showing you what this looks like so you can understand it. As we count the carbons around the ring, glucose is a hexose, meaning it has 6 carbons. The numbering starts from carbon 1. That's the carbon that has the double bond to it. And then it just proceeds around the ring. All right? All right. So anomers. Let's bring the sugars back because the experiment's over. Yeah. Okay. Yes. All right. Okay. I think this. No. Is this going to work? I think this is working. All right. The experiment worked amazingly. There we go. Just get myself oriented. Perfect. That actually worked. So if you notice here in these two sugar molecules, let me get them all the bonds oriented just the same. I'll get all the bonds oriented just the same for you. Yeah, here we go. So both of these are glucose, but they look a little different, don't they? One of them has the oxygen with the green ball, the hydroxyl group. It's sort of in plane with the ring. And the other one on this side has the hydroxyl groups pointing down, doesn't it? All right. This is alpha glucose. This is beta glucose. Glucose is able to interconvert between alpha and beta when it's just a single simple sugar that is in solution, what's called a reducing sugar. It goes in this equilibrium that you see over here. Alpha glucose opens up in the exact way that you guys were doing. And then it's an open chain with the carbonyl group, the aldehyde group, And then that aldehyde will attack that carbon. Again, depending on the side that it attacks it from, it'll either be alpha glucose or beta glucose. This is super important because polymers of alpha glucose are what make up starch that your body can utilize, that most life on Earth can utilize. Polymers of beta glucose make wood and chitin and things that aren't digestible. All right, there we go. Whew.